Um, so yeah, I'm Nathan LaPierre, and I'm a PhD student in Eliezer Eskin's lab in the computer science department. And thanks for having me here. And I'm going to be talking about our method, MsCAVIAR, for identifying causal variance by fine mapping across multiple studies. So just to define those terms, I guess this is a broad audience, but I kind of assumed basic knowledge, basically that people know that in genome-wide association studies, we've successfully established a number of associations between SNPs and complex traits. So in GWAS, we get a bunch of loci, of genetic loci that are associated with complex traits. And within each of these loci, there's a bunch of SNPs that are associated with those traits. And an important question is which of these SNPs are causal, and I'll explain in what sense we mean causal. So you can see by the scare quotes there. Most of the SNPs that are associated with the trait are not causal. They're actually just correlated with the causal SNPs through linkage disequilibrium (LD). So this complicates our ability to identify these causal variants. So usually expensive follow-up functional studies have to be performed to figure out which SNPs are causal, and that can be just cost prohibitive with so many associated variants. So we want to figure out what these causal SNPs are. Obviously, the most naive approach to figuring out what these SNPs are is to just take the top X associated SNPs and say these are the causal SNPs. And that has a couple of problems. One problem is you don't necessarily know a priori how many of these SNPs you should pick. And a second problem is that, as you can see here, you can have situations like this where the most significantly associated variants are not actually the causal variants. They're actually just highly correlated or an LD with the truly causal variants. I assume when I'm showing when I show my mouse, you can see it. So the problem of fine mapping is essentially trying to deconvolute this LD structure and to try to identify the causal variants, by which we mean the variants causing the signal of association at the locus. And there's increasing interest in performing these fine mapping studies across multiple studies. Of particular interest is transethnic fine mapping, where we have GWAS statistics populations with different ethnic backgrounds. And one of the big advantages of these of this approach is that these different populations have different correlation patterns or different LD patterns. And the reason that helps, you can imagine, is you may have two SNPs are very highly correlated and very high LD in one population, but much less correlated in a different population. So that can help you rule out a lot more potential SNPs. And so in MsCAVIAR, just to give an overview, so this is extending a popular framework, Caviar, in fine mapping in single studies to multiple studies. And as input, we take our GWAS association statistics, or Z-scores, in each study for the same set of SNPs, and we take in the LD matrices as well, or the correlation matrices. And our goal is to return a set of SNPs that, with at least rho % probability, some user set probability threshold, contains all the causal SNPs. So usually this will be something like 95. We return a set that with 95% probability contains all the causal SNPs. And what this is sort of showing the advantage of doing this over a naive approach of, say, just running a meta-analysis on these two studies is a meta-analysis yields many significant SNPs. So again, if you had to perform functional validation on each of these SNPs, it would be very costly. And here we're showing in this toy example, we return 3 SNPs, 2 of which are actually causal, because usually you're not going to be able to narrow down the set perfectly. So again, we're trying to return a set that contains all the causal SNPs. Doesn't necessarily mean that every SNP we'll return will be causal. And some of the advantages of this study is it allows for multiple causal SNPs at a locus. A lot of methods don't actually allow this. It only requires summary statistics, so it only requires association statistics, not individual genotype data, which often isn't shared for privacy reasons. And we are able to, while we assume that the causal SNPs are the same in different studies, we are able to account for potential heterogeneity in the effect sizes of those causal SNPs. So that's the approach basically, and I'm going to talk about the model a little bit. But what I'm going to try to do is, without getting into a lot of the gritty details, try to just give a sort of intuitive overview and a toy example that kind of gives you an idea of how it works. So first, I'm going to start with the Caviar model, upon which this is based for a single study. And first, I'm going to define something called a causal status indicator vector. So this vector is just a vector of indicator variables. So there's one variable for each SNP in a locus, and the variable is 1 if that SNP is causal and 0 if that SNP is not causal. So for a particular causal status vector C star, we want to determine the likelihood that the set of SNPs that are causal based on this vector, we want to determine the likelihood of that based on the association statistics we observe. And we can do this through Bayes' formula. So the probability of these SNPs being causal based on our association statistics is the probability of the association statistics given the SNPs we assume to be causal times a prior on those SNPs being causal over the sum overall possible causal statuses. And we want to return a minimal sized set of SNPs that meets the user set posterior probability threshold row. So the smallest number of ones in this vector C that gets this to at least that row threshold. Because again, we want to avoid returning a bunch of SNPs that aren't causal in our causal set. So the reason I've put this likelihood here in a red box is that's kind of the interesting part that I'm going to talk about for the rest of at least the method section of this talk, because this is really our generative model for the data. SNPs are causal. How likely is it that we observe the association statistics that we see? This prior is very simple. It's basically just that we assume each SNP has some prior probability, which we call gamma, of being causal. And so it's just a bunch of Bernoulli trials, basically. And some people are interested in functional genomics and integrating functional priors. Just as an aside, the way you would do that is you could modify this prior so different SNPs have a different prior probability of being causal. But that's pretty simple and uninteresting prior. So the rest of the talk, I'm going to give the rest of the methods section here, I'm going to give a toy example to kind of try to illustrate on a high level how we do this likelihood.
Can I ask a quick question?
Sure.
For the multi-study example that you're giving from the previous slide, actually, are you assuming that each study individually has the same ancestry, but then you're combining together studies from multiple ancestries? Do you see what I'm saying?
Yeah.
So you have to model the LD structure explicitly for within each study for a specific ancestry. Is that the idea?
Yeah. So this LD. So yeah, and the LD, of course, because if you're getting summary statistics, you may not even be able to calculate the in-sample LD. So in general, we're assuming that we just know this study is from an East Asian population, and you could, if you have the genotype data, you can calculate the LD, but otherwise you would get it from a reference panel.
Okay, thanks.
Does that answer the question?
Yeah,
Or?
yeah.
Okay.
So is
Okay.
the
Oh
fundamental
yeah.
assumption here that you've assumed a lot of the causal SNPs are shared across those racial groups? So that's why when you do meta-analysis, you could gain power.
Yeah, so we do make that assumption. I've seen some recent studies that suggest that is often the case. Of course, it may not always be the case. Hopefully the idea is, for instance, if you just have one causal SNP at a locus and it's causal in one study and not in the other, or sorry, causal in one population, not the other, you'll have a QTL, you'll have a locus in one population and you'll just have no signal at that locus in a different population. So hopefully you could tell that, but there could be scenarios where like 3 SNPs are causal in one population and 2 are causal in the other, and you wouldn't necessarily be able to tell. So yeah, unfortunately, that's just not a case we handle right now. We assume that the causal SNPs are the same in both populations.
Okay, thank you.
Yep. Okay. Okay, so I'm going to talk about this this likelihood here. So again, the probability, given that we assume certain SNPs are causal, that we observe our association statistics. Okay, and again, this is just a very simple toy example. So let's say we observe z-scores below for 3 SNPs. Again, this is the Caviar model, so we're just looking at a single study. So we see 9.9 for SNP1, 5.2 for SNP2, and 7.7 for SNP3. And say we want to test the likelihood that SNP1 is causal and SNP2 or 3 are not. So then our causal status indicator vector is going to be 1 for SNP1 and 0 for SNPs2 and 3. And so we expect that the causal SNP has some effect on the trait, which I'm going to call lambda. This lambda is actually a function of the effect size, beta, and the heritability of the trait, and also the sample size. But I'm just going to refer it as the effect size for simplicity. So we expect it to have this some effect on the trait. And for the sake of a simple example, I'm just going to say this effect size is 10. And so we have these association statistics, this causal status vector we want to test, and a causal SNP effect size. Then we expect that the effects of the other SNPs on the trait are only due to their LD with the causal SNP. So if we say that the LD between SNPs 1 and 2 is 0.5, and the LD is 3.8, then the effect size or the association, well, I'll say the effect size for SNP2 is going to be the effect size for SNP1 times the LD between SNPs 1 and 2, so 10 times 0.5 is 5. And likewise, the effect size for SNP3 will be the effect size of SNP1 times the LD between SNPs 1 and 3, so 10 times 0.8 is 8. And then we observe, the observed association statistics S are expected to be centered around these effect sizes with some covariance. And basically, in this toy example I've given, we can see that this causal status vector is a good fit to our data. Our statistics are assumed to be centered around 10, 5, 8 in this multivariate normal distribution. And in reality, they're 9.9, 5.2, 7.7. So it's a pretty good fit for the data. However, you can imagine if we had instead said, what if SNP2 is causal? Well, then lambda 2 would be 10, and lambda 1 would be 0.5, and lambda 3 would be lambda 2 times the LD between SNPs 2 and 3. So you can imagine this would be centered, this would be centered around 5, 10, and something else here. So it'd be a much worse fit for our data. So this is basically just trying to give an intuition for how, based on certain SNPs being assumed causal, how we model how likely our data is based on that. And the MsCAVIAR extension to this extends this model to multiple studies. As I mentioned before, we do assume that the causal SNPs are the same across multiple studies, but we we allow for different populations to have different causal effect sizes called heterogeneity. And we use what's called a random effects model to model this. So first we draw in the same fashion this lambda parameter, and we call this, you can think of this sort of as a global mean effect size over all possible populations. And then around this global mean, we draw the effect size of that SNP in each study I. We draw around that mean with a variance that we call our heterogeneity parameter, tau squared. And to see what that looks like in our little toy example, so now we'd have association statistics for 2 different studies. So we have these are the same 3 SNPs, and we have these association statistics. And these can be different because just because they have different LD patterns, but these can also be different because the causal SNPs have different underlying causal effect sizes. So how do we take into account both of those things? So again, let's test that that only SNP1 is causal, and assume we have the same lambda as before, 10, and so our vector is 1, 0, 0. So now we draw the effect size for each study according to the following. So lambda 1 in the Euro populate for SNP1 in the European population is drawn around this global mean with this heterogeneity parameter tau squared, and the effect size for SNP1 in the Asian population is drawn again around the global mean with this variance tau squared. And then the rest of the data is independent conditioned on these. So for example, the effect size of a non-causal SNP, SNP2 in the European population is the effect size of SNP1 in the European population times the LD in the European population of SNPs 1 and 2. So this is basically how we're able to take into account both the fact that you can have a heterogeneity in the SNP effect size and the different LD patterns in each individual study. And so just to summarize or to wrap this up, the methods section before I move on to the results. Again, we're taking as input the z-scores, the summary statistics, and the LD for the same SNPs in multiple studies. And then we want to output a causal set that with rho % probability contains all the causal SNPs. Again, this does not mean that every SNP in the set we return is causal, but so the set we return causal SNPs with this probability. And we use this Bayesian multivariate normal framework similar to Caviar that I kind of illustrated, but on top of that, we have this random effects model that allows us to account for the heterogeneity of effect sizes in different studies. And so just to quickly go over some of our results, we ran some simulation results. We picked a locus with about 50 SNPs. And again, we wanted to look at this scenario where that won't pop up a lot in reality, which is that people have these summary statistics available, but they aren't able to calculate the in-sample LD. So we take the LD matrices from 1,000 genomes, basically reference PANA LD for European and East Asian populations. And then we randomly implanted 1 to 3 causal variants at this locus and simulated their effect size, and then generated the association statistics based on that. And we did 1,000 trials with 1 causal SNP, 1,000 trials with 2 causal SNPs, 1,000 trials with 3 causal SNPs. And what this is showing here is that posterior probability threshold I talked about, rho, it turns out all the methods we tested here basically have that parameter, and we set it to 95%. So this dashed line here is for sensitivity 95%, meaning the methods are considered well calibrated if they return at least if they recover at least 95% of the causal SNPs, because that's what the model is saying it will do. And these are the methods we compared. Again, Caviar on just the separate populations individually, MsCAVIAR, the transethnic mode of Painter, which is another method, and Susie is a method we are asked to compare to, but I want to note that their method is a little different. They don't assume, or they do not attempt necessarily to return all causal SNPs at a locus. So that's why it's not well calibrated for sequels or two causal variants or three causal variants. But it's just a different model. But apart from that, all the methods you can see are well calibrated, which is good because they've been that's what they're made to do. But where the transethnic information comes in helpful is in again improving fine mapping resolution, so returning less of these non-causal SNPs, because conditioned on your method being well calibrated, you want to return as few causal SNPs as possible, so less costly functional validation has to be done. And the transethnic methods, MsCAVIAR and Painter, again, this is where the transethnic information comes in helpful. They're able to remove more SNPs from the causal set while still staying well calibrated. And we can see MsCAVIAR returns smaller set sizes on average than Painter in these simulations on average. And we believe this is due to our explicit modeling of the heterogeneity and effect sizes, which Painter doesn't do in the same way.
And so Nathan, I have a question. So what's the relationship between your last slide where the y-axis is the sensitivity and this slide where the y-axis is the set size? So how did you measure the similarity in your last slide?
So these are the same, basically these are the same simulations, but here we're just showing that the methods recovered the causal variance 95% of the time, which is the goal of the methods. And here we're showing when they recovered those causal variants, what were the set sizes they returned. So these sets they're returning. So let's say there's three causal variants, and these sets contain all of them. But Caviar is giving you 22 or 20 SNPs in that set on average. Painter is giving you 20 SNPs in that set. MsCAVIAR is giving you, I guess, 17, let's say 17 SNPs in that set. So basically, conditioned on you're returning you're capturing all the causal SNPs, you want to have as small a set size as possible. So you're not, because it would be trivial, right, to just return all the right, then you would have 100% sensitivity, but it wouldn't be very useful.
I understand that.
So you're kind of trying to narrow down the set as much as possible. Yeah.
Right. So in other words, if you also show the specificity in addition to sensitivity, you should reflect that MsCAVIAR has better specificity than other methods, right?
Right. That would be another. Yeah. Right. You could reformulate this as specificity. Yeah.
Okay. Thank you.
And then just real quickly, we did some real data analysis, a trans biobank analysis of type 2 diabetes. So we took summary statistics for GWAS on type 2 diabetes from the UK Biobank using white British people only, and from Biobank Japan. And the sample sizes for these two studies were 360,000 people and 190,000 people respectively. And we picked loci based on having strong enough peak and enough other strongly associated SNPs. And what I mean by that basically is you can imagine if you have one really strongly associated SNP and then no other SNPs that have even marginal significance, you wouldn't really need to do fine mapping. So we kind of tried to take loci that were realistic for fine mapping in that sense. And of course, for the real data, we don't know the ground truth, so we can't measure the sensitivity, but we basically rely on the simulations to show that the methods are well calibrated. And what this plot is basically showing is that MsCAVIAR returned somewhat fewer SNPs in its causal sets on average.
So just one question here. What is the overlap like between the results of these methods? Like, are they returning the same ones?
Yeah, so usually one is a subset of the other, although that's not always like if one set is smaller, it's usually a subset of another of like if MsCAVIAR returns 5 SNPs and Caviar returns 10, the 5 is usually a subset of the 10, although that's not always the case. But I don't have any hard numbers on that. Okay, and yeah, so that's all. And I want to thank my co-authors. Cody is my co-first author on this project. I want to thank Helen, Rosemary, and Farhad for their help, and Eliezer, my advisor. And we have our paper up on bioRxiv. And thank you very much.